The first stage of energy harvesting, glycolysis, literally means sugar splitting, which is appropriate because our starting molecule is the sugar glucose. During the first stage of glycolysis, one molecule of glucose has to be prepared for energy to be ready to be released. ATP has to be used rather than synthesized in two of the first steps of glycolysis so that the relatively stable glucose can be put in the form of a less stable sugar. This sugar is then split in half. The two molecules formed have 3 carbons each, whereas the starting glucose had 6 carbons.
Once the split is accomplished, the steps of glycolysis repeat themselves. It is like taking one long piece of cloth, dividing it into two, then proceeding to do the same things to both of the resulting pieces. Glycolysis accomplishes three valuable things in energy harvesting. It yields two ATP molecules and two energized molecules of NADH and results in two molecules of pyruvic acid.
After glycolysis is the final step of cellular respiration, the production of ATP molecules. There's 34 ATP molecules produced through the electron transport chain or oxidative phosphorylation, the third stage of aerobic energy harvesting. Glycolysis took place in the cytosol, and the Krebs cycle took place in the inner compartment of the mitochondria. Now, however, we move to the cristae, or the inner membrane, which is the site of the ETC. The links in the transport chain are a series of molecules. NADH and FADH2 carry off the electrons derived from glycolysis in the Krebs cycle. These are the two molecules that were oxidized and reduced earlier. The site of the ETC is the destination of these electron carriers and their cargo. Upon reaching the mitochondrial membrane, the electron carriers donate electrons and hydrogen ions to the ETC. Here is a picture of the electron transport chain. NADH has electrons at a higher energy level. Thus, when the NADH runs into the appropriate enzyme in the ETC, NADH is oxidized, donating its electrons and proton to the ETC. This process then is simply repeated down the whole ETC, each carrier donating electrons to the next electron carrier in line. The carrier is reduced by receiving these electrons, donating them to the next carrier, and then returns to an oxidized state. Each carrier thus alternates between oxidized and reduced states. You can see the carriers in the ETC. There are 3 large enzyme complexes, these 3, with 2 smaller molecules that link them. When NADH arrives in the inner membrane and bumps into the ETC's first carrier, right through 1, and then it donates electrons to it, this carrier then donates these electrons to the next carrier, 2, and this character donates these electrons to the next one, and so on down until you get to the last one, where you have protons on one side of or H ions on one side of the membrane. Then the H ions travel through the ATP synthase, which then forms with ADP and a phosphate group and creates ATP.